Hello, everybody. So this is a video on photosynthesis. I'm sorry, you don't mind my voice. I'm extremely exhausted, but I just deemed it fit to make this video for you to support your preparation for the exam tomorrow. So photosynthesis, the name photo means light. Synthesis means production. So production of food using light as a source of energy. This is what photosynthesis means. So what do we need to understand in this lesson? Focus question is, what do lettuce plant need to survive? The lettuce plant, what do they actually need to survive? Like before in respiration, we asked you, what do you need to break down the lettuce plant? Or what nutrient do you get from lettuce plant? But the lettuce plant itself, it needs to survive. What do they actually need for their survival? So among the key new vocabulary, which we're supposed to understand in this section is thylakoid, granum, stroma, pigment, NADP+. This is not the same with NAD+. NADP+, carbon cycle, and rubisco. Carbohydrate is an organic compound which contains only carbon, hydrogen, and oxygen. So with carbohydrate is contained of carbon, hydrogen, and oxygen, and is usually in a ratio 1 to 2 to 1. So that is to say you have one carbon atom with two hydrogen atoms with one oxygen atom or compound. So it depends on how you want to say it. So in generally, photosynthesis is a process by which autotrophs make organic compounds. What are autotrophs? Autotrophs are organisms that have the capacity to make their own food by themselves. So they are organisms that have the capacity to produce their own food. And they use different ways, but we're going to talk about using light as a source of energy here to make their own food. Photosynthesis converts light energy into chemical energy. So the light energy is being absorbed from the light, from the sunlight, and also some other sources of light, and it's been actually used to convert the light energy, which has been stored, is converted to chemical energy, which is needed for the plant to survive. So the products and reactant of photosynthesis are identified in the overall chemical reaction for photosynthesis. The meaning is photosynthesis equation is being written in a form of chemical reaction with 6CO2 plus 6H2O in the presence of light as a source of energy. It produces C6H12O6 plus 6O2. So if you could still remember, I told you in the beginning that respiration is a reverse reaction of photosynthesis. So you would see the product of respiration is a reactant of photosynthesis. Now, let's talk about the phases in which photosynthesis is involved in. Photosynthesis happens in two phases. One of these phases is called the light-dependent phase. So it depends on light. So that is why you call it light-dependent phase. So this phase, light energy is captured and is converted into chemical energy. So this is a unique thing that happens in the light-dependent phase. While in the light-independent phase, molecules which are formed, the chemical energy, the molecules which are produced in the light-dependent phase are being used to make glucose. So I want you to understand these two phases now. The light-dependent phase, the light energy is being captured and it's been used to produce chemical energy. And in the light-independent phase, so the chemical energy which is being produced in the light-dependent phase would be used to produce glucose, which the plant actually needs to survive. So let's go deeper into the light reaction phase. In a light reaction phase, there is absorption of light, which is the first step in photosynthesis. If there is no light source, photosynthesis will not happen. So the main thing which is needed for the process of photosynthesis is light absorption. Once the light energy is been captured, two energy storage molecules, ATP and NADPH, are being produced. Mind you, this is not NADP. This is not NADH, sorry. This is NADPH. P, to let you know, it's the one involved in photosynthesis. So NADPH are being produced and ATP. So what happens is they are very essential in the process of transporting and storage of electron, which we will see in the future. Chloroplasts are organelles that help to capture the light itself. So chloroplasts are the ones that capture the light energy. They contain two compartments, which are the thylakoid and the stroma. So I want you to understand now, chloroplast is one of the organelles which is found in a plant cell. And this chloroplast is green because of chlorophyll. And this chloroplast is divided into two compartments, thylakoid, which is a flattened sac-like membrane. So it looks like a sac. So it's a flattened sac-like membranes. So stacks of this thylakoid is called grana. If you want to singularize it, you call it granum. And stroma is the fluid-filled space which is outside the grana. So the stroma is a fluid-filled space outside the grana. So please remember this. The two main compartments of chloroplast is the thylakoid and the stroma. Now this picture will give you a better understanding of what I'm talking about here. So this picture is photosynthesis occurs inside the pigmented organelles called the chloroplast. So let's say you dissect the leaf now this way. This section here is what you see here. So then this is a leaf tissue. Then if you view this under the microscope, you will see the chloroplast inside this section here. So if you expand it, you will see the chloroplast is got the, if you expand the plant cell here, you will see the cell wall, the vacuole, the chloroplast, the mitochondria, the nucleus, the Golgi apparatus. All of these organelles are found in the plant cell. Now we want to take a look at the main actor in photosynthesis, which is the chloroplast. So the chloroplast, as you can see, is being enlarged, and we can see is made up of membrane. This is the outer membrane, and this is the inner membrane. It's a double membrane organelle. So, and we got the granum. As I said to you, the granum is a stack. It's a combination of being stack of thylakoid being put on top of each other. So the stroma is actually the liquid area, the space in between the thylakoids. So the thylakoid space located in phase one. So please know this very well. That will give you a better understanding when you want to talk about what happens in it. Now, let's talk a little bit about the pigment and pigmentation. As you know, pigments, which means what gives something its color, is called a pigment. So light-absorbing colored molecules are called pigment. I told you in the class, the pigment which is involved in the absorption of light is the chlorophyll A, the chlorophyll B, and the carotenoids. So these are found in the thalacod membrane. So different pigments absorb specific wavelengths of light. Now I want you to take a look at this diagram very quickly. You will see this is a percentage of light being absorbed out of 100 from 0. So what happens is we have three pigments here: chlorophyll A, chlorophyll B, and carotenoids. So the chlorophyll A would absorb the purple light, then it's going to drop drastically at wavelength 450 and would not absorb much of the light green and the green light. And it's going to remain down until it gets to the purple light. It goes up again, or the carrot coloration. Then the chlorophyll B also does the same thing. It absorbs a lot of the purple light, and once it gets to the green section, it drops down. The meaning is they refuse to absorb the green light. So when the green light is not absorbed, then it gets reflected. And this is exactly what gives the plant its green color. So because it's not absorbed, because the green color is not absorbed, then it is actually being reflected. The carotenoids also does very well in the light green and also in the purple area. But once it gets to the green, it drops down also. But you will see the carotenoids could still do better compared to chlorophyll A, chlorophyll B. So that's the reason why there is no light, the sunlight in the cold areas, the global areas like Sweden, like Canada. You will see the leaves turn purple. The light reflected then is the carotenoids. So the chlorophyll, the green color breaks down because there's no light source. So that makes the leaf to become yellowish, or they becomes carrot in color. So this is exactly what this section was telling you. Now let's talk about the electron transportation. Electron transport is what we talked about, the almost similar thing to what happens in the respiration. So, but this happens in a different place. So the thalacod membrane has a large surface area, providing space for a large number of electron transporting molecules and two types of protein complexes called the photosystems. This, you should understand this, that the thalacod membrane has a large surface area. It needs to have this to accommodate the electron transport molecules and also two types of protein complexes, which are called the photosystems. Photosystems contain the light capturing pigment and proteins that play important roles in light reaction. So photosystem contain the light capturing pigment, all of the light capturing pigment which we need in the process of photosynthesis is found in either photosystem 1 or photosystem 2. And also, there are special proteins which are important in playing several important roles in the light reaction. So, electron transport, light energy excites electron. So, if the light energy strikes the water molecule in photosystem 2, the water molecule gets splitted, and that would activate the electron, and makes them very active. So next, the excited electrons will move from the photosystem 2 to an electron acceptor molecule. So these electron acceptor molecules would move the electron from photosystem 2 to photosystem 1. So now the electron acceptor molecules would transfer the electrons which are being excited in a photosystem 2 along a series of electron carriers. So there are several electron carriers to photosystem 1. Photosystem 1 would transfer the electron to a special protein called ferredoxin. So the ferredoxin would then transfer the electron to the electron carrier, which is NADP+. So NADP+ would not get reduced because once it has access to electrons, it forms an energy storage molecule, which is NADPH. It was actually oxidized from NADPH to NADP+. So once it has access to electron again, it would get reduced quickly to form NADPH. Chemosmosis is a process by which ATP is actually being produced. So what happens in chemiosmosis is is an osmosis whereby chemicals are actually involved in it. Hydrogen ion and some other charged molecules are involved in it. So chemiosmosis is a mechanism by which ATP is produced as a result of flow of electrons. So it's a process of producing ATP when the electrons actually, when the flow. So when the electrons flow, they produce ATP. We're going to talk about that later. But if you understood when I talked about the chemiosmosis in cellular respiration, so once the electrons once they flow from one protein to another one, so they would allow electrons, hydrogen ions to migrate. So across the thylakoid membrane. So they keep on migrating across the thylakoid membrane until they become so much on one side of it. Then what happens is the ATP synthase would allow the hydrogen ions to go across to cross down the electron gradient. So whenever it crosses down the electron gradient, that would produce as many ATP as you want. So the breakdown of water is essential in this process. So now let's talk about the second phase, which is the carbon cycle. Mind you, all of the electrons you gathered in the first process, which is light reaction, and all of the electron carriers, electron storage, and all of this stuff, they actually been doing all of these things to prepare themselves for the light independent reaction, which is called the carbon cycle. So the second phase of photosynthesis, called the carbon cycle, energy is stored in organic molecules such as glucose. So I will start from the first thing here, which is 6CO2, which is called carbon fixation. So carbon molecule is being fixed into the reaction process. So what happens is we have because we don't really have up to 6 carbon compounds. So five carbon compounds was brought up for the reaction process. So Rubisco, which is an enzyme, would facilitate the combination of one CO2 molecule, which is going to bring in carbon atom to the reaction. So that gives you a total number of 6 carbon atoms times 6. So that gives you 36 carbon atoms. And that's why we had 12 3-PGA. So the 3-PGA molecule is a very high energy molecules. So we need to it needs to use 12 ATPs. It also needs to use 12 NADPH to change the 3-PGA to a higher energy molecule. So it needs to take ATP and it needs to take NADPH because the electrons which is being carried with NADPH are being used. And the ATP, which is going to give it more energy to become a high energy molecule, so would be given to it. So now from 12,3-PGA, then you have 12G3P. So from 12G3P, 2 molecules of this 12G3P will be taken off at step 3 to produce glucose, fructose, and starch. So mind you, when 2 has been taken off, so you are left with 6-ribulose 5-phosphate. So the 6-ribulose phosphate would take in 6 ATP to change the 6-ribulose phosphate to 6-ribulose 1,5-bisphosphate. So the reaction will start all over again. So the main thing is you have 4 steps in carbon cycle. The first one is carbon fixation, where carbon dioxide is being added to the 5 carbon compounds to make it 6. So the second stage is the production of 3-PGA, 12 molecules of 3-PGA. So because this 3-PGA needs to be changed to a very higher energy molecule, so it uses 12 ATP and 12 NADPH to change it to a higher energy molecule, which is the G3P, 12 molecules of it. So two compounds of this 12 12 molecules will be used to produce glucose, fructose, and starch. So the remaining 10 will proceed into the reaction. So that gives you 5 carbon atoms. So, but it's actually 6 molecules of it. So you have 6 molecules of ribulose, five phosphate. So once you add 6 ATP, then it changes that ribulose 1 ribulose phosphate. It changes it to 6 ribulose 1,5 bisphosphate. So now let's explain in more detail. The first step of carbon cycle is called carbon fixation, which CO2 molecules combine with the 5 carbon molecules to form 3 phosphoglycerate, which is the 3-PGA. Then the second step, chemical energy stored in the ATP and NADPH will be transferred to 3-PGA to form a glyceraldehyde 3-phosphate, which is a higher energy molecule. So when that happens, the third step, some of the G3P molecules will leave the cycle to be used to produce glucose and other organic compounds. So the fourth step, which is the final step, is when the enzyme, one of the most important enzyme, riboscope, would have to convert the remaining G3P molecules into 5 carbon molecules called the ribulose 1,5 bisphosphate, so RUBP. So these molecules combine with the new CO2 and the cycle would continue again. So we have some plants which would not be able to go through these normal processes we've talked about. So we call them the C4 plant. So the C4 plant's pathway, it allows the plant to maintain photosynthesis while reducing water loss. So instead of bringing 3 carbon atoms, instead of bringing, let me go back and make you see. So you could see it brought 5 carbon atoms to the reaction here, but a C4 will bring 4. So it brings 4. So why does it do this? It tries to make sure it does its activities when the light energy is not that much. So it could help. If you know the differences between the C3 and the C4, you will be able to understand why the C4 is very important. So in carbon cycle, it produces 3 carbon compounds, as you saw. So from these, you see the 3 carbon compounds here. So that's why from the 6, it breaks into 12 threes. That's why it's called C3. But a C4 will bring intermediate 4 carbon compounds. And the reason it does this, it's just for it to be able to store, save a lot of water loss. So every photosynthetic plant uses C3. We know that. So as its primary source of photosynthesis. But a C4 photosynthesis, it happens around just like, let's say 4 to 3% of vascular plants. We example are the crabgrass, sugarcane, corn. So the seafood plants have a significant structural modification and the arrange of the cells within the leaf. So the cells are arranged in a different way from the C3 plants. So that makes it easy for the plant to save water loss. While the CAM plant, which is called the crustacean acid metabolism, is found in desert plants. So these plants will collect the CO2 at night and store in organic compounds. So what does that mean? The stomata is the part of the plant which is underneath the leaf, which helps to absorb gases and let gases leave and also liquid. But the plant knows if they open the stomata in the day, they will lose a lot of water. So what do? They would collect the CO2 at night and store it in organic compounds. So during the day, the plant would release the CO2 from the organic compound for the light-dependent cycle of photosynthesis. So during the day, so they would release the CO2 within the system without opening the stomata and use it to produce a light-dependent cycle of the photosynthesis. So the CAM photosynthesis is a third type of photosynthesis that occur in arid plants that are in dry places. So now the C3 is normal. The C4 is a type, is a process by which the plants want to store some water. And also the CAM is also, the plant do not want to lose much water molecules. So this is why we call them alternative pathways. So I think this is clearer enough. I wish you all the best, and I hope this is helpful. Thank you very much for your attention.